Hey guys, we are going to be going over the monohybrid cross, basically what it is and how to do it. So a monohybrid cross, let's break down this word. Mono means one, hybrid means to mix, and cross means to mate. So basically what this talks about, or what this shows us, is the answer to the question of what is the probability, so like the likelihood. Wow, I spelled that so wrong. That's embarrassing. Okay, probability or likelihood on passing one gene, that's what the one refers to, one gene given the genotypes of a male and a female. So cross is mate. So basically if this male and female got together and had a kid, what's the probability of passing on like one of their genes or one of their traits? So mono only talks about one gene or trait from each person. So we're going to go through a little bit of a scenario. My scenario. Okay, let's take a man who is heterozygous. Let's change this up actually. Okay, a man homozygous dominant. A man homozygous dominant for a unibrow. Mates with a woman who is homozygous recessive for 2 eyebrows. Okay, so my question would be, what are the possible genotypes and phenotypes of their children? Well, let's go with child. Okay. So, in order to figure out probability, I have to complete a Punnett square. Before I do the Punnett square, that means that I need to figure out what are my genotypes of each of these people. Okay, so we have a man who is homozygous dominant. Homo means same. Dominant means I'm going to have 2 uppercase letters. I'm going to choose U. U could have been, it could have been any letter, but U for unibrow. Yay, unibrow. Okay, U. So with a woman who is homozygous recessive. Okay, homo means same, recessive means little letter, so I'm going to have 2 little u's. Okay, now I need to complete a Punnett square. So Punnett square. Now I put the genotypes of my parents on the top. So 1 letter in each box for my monohybrid cross. This is what I'm completing. Punnett squares help me complete. I'm going to actually put this down here. These are Punnett squares. This is a Punnett square. Punnett square. Punnett squares help me complete my monohybrid cross. So I have my dad on the top and my mom on the bottom. Or on the side, sorry. Alright, now to fill in this, it's like a multiplication table. So I have these big U's in each of the boxes, and I always write, like I always pick one to write first. I always write the dominant letter first, and then I follow it with my recessive. Okay, so this question asked me, what are the genotypes and phenotypes? So my genotype is really, I only have one genotype, and that is U, capital U, and lowercase u. That means that I have a heterozygous genotype. And this is 100% of my children are going to have this genotype. Now my phenotype, because it's mixed, because it's heterozygous, but I have a dominant present, that means that all of my children are going to have a unibrow. So we would definitely need to invest in tweezers. Okay, now we call this, when we have like an initial cross, this would be the parent cross. My parents were U, and U. Okay, now that was my parents, and then what we call in science kind of our first generation, that's when we cross our parents, which are normally homozygous dominant and homozygous recessive. My F1 generation was actually everything inside of this box, so that means that my F1 generation would be capital U, lowercase u. Alright, now there's something called an F2 generation. An F2 generation is what happens when I cross my F1 generation with each other. So F1 cross F1. This normally happens with plants, so if we're talking about kids, this would be like a brother and a sister had sex together. So, you know, we don't actually do that, but you can do it in plants. Okay, so what this would look like is if I set up my Punnett square, I would have my F1, one of them up here, and the other one on the side. And then I just fill this in. Okay. Alright, so now if I cross my F1 generations together, my genotypes and phenotypes for my F2 generation are actually going to be a little bit different. Alright, now we're going over this. I know this like parent, F1, F2 might be a little bit confusing, but this is what Gregor Mendel went through with his plants to actually figure out how genetics works. And you're like, who is Mendel? Mendel is the father of genetics. He basically just spent hours of his life like mating plants with each other. So he spent most of his life watching pea plants have sex with each other in order to determine all of these different relationships. So thank you, Mendel, for your pastime. Okay, so my different genotypes that I would get are, I had UU, that was homozygous. Ooh, sorry, the lights went out. Okay, there we go. I had homozygous dominant, I had heterozygous, and I had homozygous recessive. Now, what was the percentage of each of these? Well, for my homozygous dominant, that accounted for 1 out of my 4 boxes, so that was 25%. Alright, my heterozygous counted for 2 out of my 4 boxes. That means that that was 50%. This is the likelihood that this generation would end up with a unibrow or not. And then I had a 25% chance that my child would, or that these people's child would actually not have a unibrow, that they would have 2 eyebrows. So lucky for them. Hopefully that works. All right, so these percentages represent probability. I can't spell today. I really can't spell. That's probability of inheriting the trait. Now there is one more thing that I want to leave you with. Okay, one more little fun fact. Sometimes you will actually notice in families that traits can skip a generation. So that is called something called a masked trait. So this is when traits skip a generation. So you'll notice, like, if you've ever known families that have red hair, a lot of the time red hair will be like in the grandparent generation, will skip the parent, and then will be in the grandchild. So it'll be like your grandma has it and you have it, but your parent doesn't have it. That's called a masked trait. Or like oftentimes families having twins, that sometimes skips a generation. So like, again, grandparents would be twin, your parents wouldn't be, but then you would be a twin yourself. So there are just certain things that sometimes skip a generation. That's why we see like the parent F1 and F2 generation, so that we see what actually is in the genotype, and we'll actually be able to map this out a little bit later in the unit. But that was your lesson on monohybrid crosses, which you are going to do. Here were some examples provided, but you are going to answer the questions that are on the worksheet that start here, and then continue on depending on which packet that you have. But get started and have fun.